Today's topic is nervous system, and nervous system is composed mostly of the nervous tissue, which was discussed in the previous lecture. And the nervous system is subdivided anatomically by central nervous system and peripheral nervous system. Central nervous system includes brain, cerebrum, cerebellum, medulla oblongata, pons varolii, etc. Spinal cord, retina of the eye, olfactory region of the nose, consisting of the neurosensory cells, terminal and terminal. Peripheral nervous system includes peripheral and cranial nerves of ganglia, autonomic and sensory, nerve ending, sensory and effectory, motor or secretory. Gray matter is composed of neurons and neuroglial cells. It's forming cortex and nuclei, basal ganglia, and cerebrum, cerebellum, and brainstem. Horns, anterior, posterior, and intermediate within the spinal cord. And neuropilum, regions of central nervous system represented by plexuses of axon terminals, dendrites, and of neuroglia processes. White matter, the accumulation of myelinated nerve fibers and neuroglial cells, is forming tracts in brain and fasciculi within the spinal cord. Cerebrospinal fluid is produced by the choroid plexus and meninges, the dura mater, consisting of dura mater, arachnoid, and pia mater cerebri, protecting the soft tissue of the central nervous system of the brain. Here you have the main anatomical divisions of the central nervous system, including the spinal cord, medulla oblongata, pons varolii, cerebellum, cerebral cortex, diencephalon, and basal ganglia. Cerebral cortex, the neurons of cerebral cortex are responsible for the integration of sensory information and the initiation of the voluntary motor responses of these signals coming from the outer space or inner space of the organism. Cytoarchitectonics of the cerebral cortex includes 6 layers of cells, namely molecular layer, horizontal cells, external granular layer, stellate cells, external pyramidal layer consisting of small pyramidal cells, internal glandular layer consisting of stellate and small pyramidal cells, internal pyramidal or ganglionic cell layer. Here are the most remarkable cells of the cerebral cortex, the cells of beds of giant pyramidal cells, which are having axons leaving the cortex of the cerebrum and traveling to the peripheral target organs, muscular components, muscular tissue for the generation of response to the signals coming from the outer space, from the outer environment. And multiform cells layer consisting of Martinotti cells. Here we have these cells on the 6 layers, combination of the schematic view. Here you have the silver impregnation method of the bed cells, 3 bed cells located in the ganglionic cell layer. Here you have the methylene blue stained section of the cerebral cortex with giant pyramidal cells of beds located with the sharp angles direction to the molecular layer or the surface of the cortex. Here you have this increased magnification of horizontal cells, of stellate cells, of pyramidal cells, cells of beds of fusiform cells, and cells of Martinotti. Here is the histological specimen section with hematoxylin eosin staining method, showing you the blood vessels on the surface of the cerebral cortex, the molecular cell layer, the lightest, with the layer which is consisting minimal count of the neurons, outer granular layer, outer pyramidal layer, inner granular layer, inner pyramidal layer, where the cells of beds are located with their sharp angles directed towards the surface of the cerebral cortex. In multiform layer, which should be somewhere beneath the plane of section. Here you have the cells of beds on the increased magnification with axon terminals leaving the basal part, basement part of the cell, and dendritic processes, apical dendrit, lateral dendrites, forming the neuropilum and forming the synaptic junctions with the adjusting bed cells and adjusting the small and granular cells, and with the small pyramidal and stellate cells. Cerebellum. Cerebellum coordinates muscular activities throughout the body, regulation of balance and skeletal muscle tonic contractions. Sharply folded cerebellar cortex demonstrates characteristic arborvitae figures of the sagittal sections. Neurons of cerebellar cortex are organized with 3 layers of cells, which include the molecular layer, basket cells, stellate cells, dendrites of Purkinje cells. Purkinje ganglionic cell layer consists pericardia of the Purkinje cells, and granular cell layer with small granule cells, Golgi cells, cerebellar glomeruli, axons of Purkinje cells. Here you have this schematic view of the gray matter of cerebellar cortex with white matter located beneath the cortical layers. Here you have the ganglionic Purkinje cells of the second layer. Here you have molecular layer, which is housing the multiple ramifications of the Purkinje cells, and here is the granular layer, which is poorly stained or completely unstained with silver impregnation method, where the axon of the Purkinje cells are leaving the cell bodies and traveling to remote target organs of muscle tissue, muscle system. Here is increased magnification, the ramifications of the Purkinje dendrites, the stellate cell, the basket cell forming the basket conglomerates of the cell bodies of the basket processes are forming conglomerates around the cell bodies of the Purkinje cells. Here you have the granular layer consisting of the granular cells, small granular cells consisting of the Golgi cells, consisting of the glomeruli, contacting synaptic junctions between the different subsets of the neurons of the cerebellar cortex. You have the hematoxin neosine stained specimen of the cerebellar cortex with molecular cell layer, the surface covered with pia mater cerebri. You have the ganglionic cell layer of Purkinje cells. Increased magnification is depicted here. This is granular layer and white matter of the cerebellar cortex beneath the cerebellar cortex. You have increased magnification of the Purkinje cells, pear-shaped cells with dendritic processes ramifying in the molecular layer, axon terminals, and leaving the cell body, the pericardia of the Purkinje cells, and ganglionic cell layer, incomplete cell layer of the Purkinje cell pericardions of cell bodies. Spinal cord. Its cross sections of the spinal cord, white matter is peripheral and gray matter is internal, and has the general shape of letter H or butterfly. In the center of an opening, the central canal, which develops from the lumen of embryonic neural tube, is existing. This canal is continuous with the ventricles of the brain, contains cerebrospinal fluid, and is lined with ependymal glia. The gray matter forms the anterior horns, which contain motor neurons whose axons make up the ventral roots of the spinal nerves, and the posterior horns, which receive sensory fibers from the neurons located in the spinal dorsal root ganglia. Spinal cord neurons are large and multipolar, especially the motor neurons of the anterior horns. White matter consists of anterior, posterior, and lateral fasciculi tracts of myelinated nerve fibers. This is the schematized version of the structure of spinal cord. This is the view, the gray matter with butterfly configuration, the dorsal root with spinal ganglion or dorsal root ganglion, the anterior horns, posterior horns. Anterior horns are thick and lower, the posterior horns are thinner and higher, delimiting the lateral fasciculi or funiculi, anterior funiculi and posterior funiculi or fasciculi. And here you have the dorsal mediana septum and ventral mediana sulcus. Here you have the multipolar motor neuron of the anterior horns, shown in increased magnification, with Nissen subsets filling the cytoplasm, representing the endoplasmic reticulum components of the motor neuron, the prominent nucleolus, which is encompassing the active synthesis of the proteins, substances needed for the normal histophysiology of the neurocyte, and the euchromatinized nucleus. The axon terminal, which is leaving the cell body through the axonic hillock of the motor neuron. Here is the same structures on the increased magnification, which you have possibility to make designations by your own. Meninges and blood-brain barrier protection of central nervous system. The central nervous system is completely enclosed by 3 connective tissue layers called meninges. The top external dura mater, the middle arachnoid layer, and number Z, the delicate pia mater that directly contacts the neural tissue. The arachnoid layer contains much cerebrospinal fluid, which helps cushion the central nervous system organs within bony enclosure. The choroid plexus consists of elaborate folds of vascularized pia mater covered by ependyma that projects from walls of the cerebral ventricles. The water removed from capillaries is transferred into the ventricles, that the cerebrospinal fluid by the choroid plexus is produced cerebrospinal fluid. In most central nervous system regions, neurons are protected by blood-brain or hematoencephalic barrier consisting the perivascular feet of the astrocytic processes, thick basement membrane, and occluding junction between the endothelial cells of continuous capillaries. The thick dura mater consists of dense fibroelastic connective tissue that is continuous with the periosteum of the skull. Around the spinal cord, the dura mater is separated from the periosteum of the vertebra by the epidural space, which contains the plexus of thin-walled veins and loose connective tissue. The dura mater is always separated from the arachnoid by the thin subdural space. The internal surface of the old dura mater, as well as its external surface in the spinal cord, is covered by simple squamous epithelium of mesenchymal origin, which is called neurotelium. The arachnoid has 2 components: a sheet of connective tissue in contact with the dura mater, and a system of loosely arranged trabecula composed of collagen and fibroblast, continuous with the underlying pia mater. The underlying pia mater, surrounding the trabecula, is a large sponge-like cavity, the subarachnoidal space filled with the cerebrospinal fluid. This fluid-filled space helps cushion and protects the central nervous system from minor trauma. The subarachnoidal space communicates with the ventricles of the brain, where the cerebrospinal fluid is produced. In some areas, the arachnoid penetrates the dura mater and protrudes into the blood-filled vein of sinuses located within that layer. These cerebrospinal fluid-filled protrusions, which are covered by vascular endothelium cells lining the sinuses, are called arachnoid villi, which function is a site for absorption of central cerebrospinal fluid into the blood of the vein of sinuses. The innermost pia mater consists of flattened mesenchymally derived cells closely applied to entire surface of the central nervous system. The pia does not directly contact nerve cells or fibers, being separated from the neural elements by the very thin superficial layer of astrocytic processes, called the glia limitans, which adheres firmly to the pia mater. Together, the pia mater and the layer of osteocytic end feet form a physical barrier separating central nervous system from cerebrospinal fluid in the subarachnoidal space. Blood vessels penetrate the central nervous system through long perivascular spaces covered by pia mater, although the pia disappears when the blood vessels branch to form the small capillaries. However, these capillaries remain completely covered or enveloped by the perivascular limiting layer of astrocytic processes. Here you have the diagram of the meninges showing you the dura mater cerebri, subarachnoidal space, arachnoid membrane, subarachnoidal space, pia mater cerebri, and brain cortex, cerebral cortex. Here is the arachnoid with subarachnoidal space filled with the liquor, was filled with the cerebrospinal fluid. The choroid plexus consists of highly specialized tissue with elaborate folds and many villi projecting into the four large ventricles of the brain. It is found in the roof of third and fourth ventricles and in two lateral ventricle walls, all the regions on which are ependymal lining directly contacts the pia mater. Each villus of the choroid plexus contains a thin layer of well-vascularized pia mater covered by cuboidal ependymal cells. The function of the choroid plexus is to remove water from blood and release it in the cerebrospinal fluid, which is produced continuously up to 500 milliliters per day, and completely filling the ventricles, the central canal of the spinal cord, and subarachnoidal and perivascular spaces. Cerebrospinal fluid provides the ions required for central nervous system neuronal activity, maintains intracranial pressure, and in the choroid arachnoid serves to help absorb mechanical shocks. Adult human contains from 100 to 150 milliliters of cerebrospinal fluid, so the rest 300 or 350 milliliters are reabsorbed by the arachnoid processes. Choroid plexus, which is blood vessels covered with the ependymal glia, cuboidal cells of ependymal glia, and here is the cerebrospinal fluid filtrated from the blood vessels of the choroid plexus. And choroid and the arachnoidal villi of arachnoidal granulation, so the arachnoidal villi are the regions where the cerebrospinal fluid is reabsorbed. Blood-brain barrier is a functional barrier that allows much tighter control than that in the most tissues over the passage of substances moving from the blood into the central nervous system tissues. The main structural components of blood-brain barrier is the capillary endothelium, in which the cells are tightly sealed together with well-developed occluding junctions and with little or no transcytosis activity. The limiting layer of perivascular astrocytic feet that completely envelopes the basal lamina of the capillaries in most central nervous system regions further regulates passage of moleculars and ions from blood to brain. The blood-brain barrier protects neurons and glia from bacterial toxins, infectious agents, and helps maintain the stable composition and constant balance of ions in the interstitial fluid that is required for normal neuronal function. The components of blood-brain barrier are not found in the choroid plexus where cerebrospinal fluid is produced. In the posterior pituitary, which releases hormones, and in regions of the hypothalamus where plasma components are monitored. Here is the schematic view of the central nervous system, demonstrating the neuron-glia relationship and formation of blood-brain barrier. Here is the blood capillary, endothelial, with the intercellular junctions, the basement membrane, the pericyte, the end feet of the astrocyte, enveloping the whole capillary surface, the pia mater cerebri, the microglial cells, which are monitoring for the antigens and playing protective role in the central nervous system organs. These are the macrophage system cells, delivering from the monocytes, the oligodendrocyte forming the myelin sheets around the axon terminals. Here you have the synaptic junction. Here you have dendrites. You have the axon process of the neuron. Here you have the ependymal glia lining the ventricles or the central canal of the spinal cord. Here is the tannicid, the element of the component of the ependymal glia, which is contacting directly the cell body of the neuron, providing the substances needed for the normal function of the neuron directly from the lumen of the ventricles of the spinal cord. Peripheral nervous system includes peripheral and cranial nerves, sensory and autonomic ganglia, sensory and effectory nerve endings, and peripheral nerve in its turn includes fasciculus or bundles of myelinated nerve fibers, and connecting tissue investments forming the endoneurium surrounding each particular myelinated nerve fiber, perineurium, connective tissue and plus epithelioid cells surrounding the bundles of the myelinated nerve fibers, and epineurium, layer of dense connective tissue surrounding the whole large nerve. Peripheral nerves establish communication between centers in the central nervous system and the sense organs and effectors, muscles, glands. They generally contain both afferent and efferent nerve fibers. Afferent fibers carry information from internal body regions and the environment to the central nervous system. Efferent fibers carry impulses from the central nervous system to the effector organs commanded by these centers. The effector organs can be either muscle or the gland. Nerves possessing only sensory fibers are called sensory nerves. Those composed of only fibers carrying impulses to the effectors are called motor nerves. Most nerves have both sensory and motor fibers and are called mixed nerves, usually also with both myelinated and unmyelinated axons. Peripheral nerves consist of axons from motor neurons in the spinal cord, sensory neurons and autonomic neurons in ganglia. All the axons are enclosed within the series of Schwann cells, but only large myelinated axons have myelin sheaths and nodes of Ranvier. From one to adjusting node of Ranvier, the impulse, nerve impulse is jumping, responsible for the increasing the speed of the transposition of the electric impulse. It's called saltatory transposition of electric impulses or the signal impulses by the nervous cells, produced by nervous cells. Endoneurium is a thin connective tissue layer immediately surrounding Schwann cells in the peripheral nerves, containing a few non-fenestrated capillaries and much reticulin. Groups of axons with Schwann cells and endoneurium are surrounded by perineurium, consisting of layered squamous fibroblastic cells joined by tight junctions to make a blood-nerve barrier. In large peripheral nerves, groups of axons are subdivided as fascicles, each of which is surrounded by perineurium. Surrounding the perineurium is a thick outermost layer of dense irregular connective tissue, which is called epineurium. So here we have similarities between the connective tissue names of in the tendons, in nerves, and in the muscular organs, including the endotendinium, peritendinium, and epitendinium, the bundles of the connective tissue in the tendon, the endomysium, perimysium, and epimysium in the muscular system, and the endoneurium, perineurium, and epineurium in the peripheral nerves. Here you have these components, announced components in the designatory marks of the diagram. Here you have the node of Ranvier. Here you have the Schwann cell myelin sheath. Here you have endoneurium surrounding each particular nerve fiber, myelinated nerve fiber. Here you have the perineurium, which is surrounding the bundle of nerve fibers. And here you have epineurium, which is surrounding the whole nerve, composing of the dense irregular connective tissue. The cross-sectioned histological specimen of the peripheral nerves, showing you the perineurium surrounding the bundles, the epineurium surrounding the whole nerve, the white adipose fat or white adipose tissue, the blood vessels, namely the arterial blood vessel and smaller size blood vessel filled with the red blood cells. And here you have the red blood cells giving the evidence that you are visualizing the blood circulatory system. Here you have the peripheral nerve in different, but by different dyes, stained by different histological dyes, hematoxin and eosin stained. Peripheral nerve with perineurium surrounding the connective tissue, soft, thin layer surrounding endoneurium. Here you use blood vessels in the perineurium. Here you have the perineurium sheath and myelinated nerve fibers surrounded with thin segments of the connective tissue. Here you have this segment of the peripheral nerve with myelin sheath surrounding the axon terminal. The dark dot is axon terminal. The myelin sheath is phyton, this clear zone which is surrounding, since the lipid-rich material was extracted due to the histological specimen production, processing. Here you have the epineurium with adipose cells and blood vessel inside, the perineurium, the endoneurium coming inside the bundle of the nerve fibers, and myelinated nerve fibers, which are depicted on the higher magnification level. The peripheral nerve electron micrographic representation on small magnification, relatively small magnification. This is the myelin sheath, myelinated nerve fiber. They're in minority. The majority is non-myelinated nerve fiber, where the Schwann cell cytoplasm is surrounding the nerve cell processes, axons of nerve cells, without making the myelin sheath. And here you have another example of the peripheral nerve shown by electron microscopic view. The reticular fibers, the perineurium, the myelin sheath, the Schwann cell nucleus, the fibroblast nucleus, the non-myelinated nerve fibers, and mitochondria in the axon terminals of the nerve, of the peripheral process of the neurocytes. Ganglia are typically ovoid structures containing neuronal cell bodies and their surrounding glial satellite cells supported by the delicate connective tissue and surrounded by a dense capsule. Because they serve as relay stations to transmit nerve impulses, at least one nerve enters and another nerve exits from each ganglion. The ganglion of the nerve impulse, the direction of the nerve impulse determines whether the ganglion will be a sensory or an autonomic ganglion. Sensory ganglia receive afferent impulses that go to the central nervous system. This is otherwise called spinal ganglia or dorsal root ganglia. Sensory ganglia are associated with both cranial nerves or cranial ganglia and the dorsal roots of the spinal nerve, spinal ganglia. The large neuronal cell bodies of ganglia are associated with thin sheet-like extensions of small glial satellite cells. Sensory ganglia are supported by a distinct connective tissue capsule and an internal framework continuous with the connective tissue layers of the nerves. The neurons of these ganglia are pseudo-unipolar and relay information from the ganglions, nerve ending to the gray matter of the spinal cord via synapses with local neurons. Here you have location of dorsal root ganglia or the spinal ganglion and the dorsal roots coming to the spinal cord, segmented structure. Here you have the spinal ganglion on the small magnification light micrographic representation and high magnification of the pseudo-unipolar neurons surrounded with satellite cells. The groups of pseudo-unipolar neurons are located beneath the capsula. The capsula includes adipose tissue, white fat, and fasciculi of the nerves are traveling in the central position of the spinal ganglion. Here is another example of the spinal ganglion from your lab guide with designatory arrows, which are showing you the ventral root, the dorsal root, the pseudo-unipolar neurons with subcapsular location surrounded with the satellite cells with central location of the nerve fibers. And the somatic and autonomic nervous system definition. The somatic nervous system is associated with the voluntary control of the body movements via the use of skeletal muscles. It is responsible for the functions we are aware and can consciously influence, including the movements of our arms, legs, and other parts of the body. The somatic nervous system consists of both afferent sensory and efferent motor nerves. It is also responsible for the reflex arc, which involves the use of interneurons to perform reflexive actions. The autonomic nervous system, formerly referred to as the visceral or the vegetative nervous system, is a division of the peripheral nervous system that supplies muscle and glands, and thus influences the function of the internal organs. Sympathetic and parasympathetic subdivisions. The autonomic nervous system is the control system that acts largely unconsciously and regulates bodily functions such as the heart rate, beating, digestion, respiratory rate, pupillary response of the eyeball pupil, urination, and sexual arousal. This system includes sympathetic and parasympathetic subdivisions. Neural cell bodies of preganglionic sympathetic nerves are located in the thoracic or lumbar segments of the spinal cord, while those of the parasympathetic division are in the medulla and midbrain and in the sacral portion of the spinal cord. So this is called the preganglionic sympathetic nerves are called thoracolumbar segment. And, parasympathetic centers are located in cerebral sacral portions of the spinal cord. Autonomic nerves use two neuron circuits. The first neuron of the chain with the preganglionic fiber is located in the central nervous system. Each axon forms a synapse with the postganglionic fibers and the second multiple neuron in the chain located in the peripheral ganglion system. The chemical mediators present in the synaptic vesicles of all preganglionic axons is acetylcholine. Here you have the somatic and visceral reflex arcs. In the somatic reflex arc, showing you the blue coloration is showing you afferent neuron processes within the spinal nerve cell bodies located within the spinal ganglion, and axon receiving the dorsal horns of the spinal cord. Here it's forming the synaptic junction with the interneuron, and then the effectory motor neuron is sending the impulse to the target organ, for example, in the skeletal muscle fibers, in order to remove the fingertip from the source of pain. It's shown here. And visceral reflex arc is continuous with the consisting of the blue coloration of spinal root, dorsal root ganglion, making the synaptic junction within the interneuron, within the medial horns of the spinal cord, contacting the second neuron or preganglionic neuron, sending its process to the prevertebral ganglion of the autonomic system, or to the sympathetic chain ganglion, or the sympathetic nervous system, and entering the with signal electric impulse coming through the axon of the second ganglion of the motor or effector nerve, entering the gut and giving command either to contraction of the smooth muscle cells of the gut, of the giving command of the secretory activity of the glands present in the digestive tube. So it consists of the two neurons circuits in the effectory nerves. Autonomic ganglia are small bulbous dilatations of the autonomic nerves, usually with multipolar neurons. Some are located within the certain organs, especially in the walls of the digestive tract, where they constitute the intramural ganglia. Autonomic nervous system has two parts, sympathetic and parasympathetic divisions. Sympathetic second neurons are located in small ganglia along the vertebral column, while second neurons of parasympathetic series are located in very small ganglia, always near the or within the effector organs, for example, the wall of the stomach and intestine. Due to a huge number of neurons in ganglia innervating the gastrointestinal tube, they are currently treated as metasympathetic or enteric division of autonomic nervous system. Enteric ganglia lack distinct capsules. Pericardia associated satellite cells surrounding connective tissue form plexuses instead of ganglia. For example, submucosal plexus of Meissner and mesenteric plexus of Auerbach, supporting the gastrointestinal tube. And it is noteworthy to remember that there are specific cells responsible for the peristaltic contraction, giving the pacemaker, so-called interstitial cells of Cajal, which are treated as the pacemaker cells of the peristaltic junctions of the peristaltic movements of the gastrointestinal tube. You have view on the autonomic ganglion with multipolar neurons located in the connective tissue investment. Then you have the enteric plexus consisting of the nerve cell bodies surrounded by the satellite cells and surrounded with the fibers and fibroblasts, forming the enteric plexus in the enteric or submucosal layer of the gastrointestinal tube. Here is the diagram of the multipolar neuron with axon, dendrites, and synaptic junctions. Axons forming terminal boutons. Long process is axon. The short ramified process are dendrites. And the synaptic junctions are shown here. We have the axon-dendritic junction, axon-somatic junction or synapse. And here you have the axon-spinose, dendritic spinose synapse. So axon and dendrites, we discussed this topic in the nerve tissue segment of the lecture course. But I'm just emphasizing that it was previously believed that axons bringing the information from the cell to the periphery, and dendrites bringing information from the periphery to the cells, to the nerve cell bodies. But now it is a little bit, another treatment has in with practical treatment that axons can deliver segment in both sides, from the periphery to the nerve cell somatic part of the body of the nerve cell, and from the nerve cell body to the periphery. So it depends that the length of the process should have been denominated as the axon or dendrites, not the direction of the signal transduction is giving the name of the nerve cell process. Axon is a long slender projection of the nerve cell that typically conducts electrical impulses known as the action potential away from the nerve cell body. But this is as a rule. The function of the neuron is to transmit information to different neurons, muscles, and glands. This is as a rule. But there is exclusions in this rule. In certain sensory, namely pseudo-unipolar neurons, such as those of touch and warmth, the axons are called afferent nerve fibers, and the electrical impulse travels along this from the periphery to the cell body, and from the cell body to the spinal cord along another branch of the same axon in the pseudo-unipolar neurons. Dendrites or dendrons are branched cytoplasmic extensions of the nerve cell that propagate the electrical stimulation received from the other neural cell to the cell body of the neuron from which the dendrites project. Electrical stimulation is transmitted onto dendrites by upstream neurons, usually via the axons, via synapses, which are located at the various points throughout the dendritic tree. Here you see the diagram of sensory nerve endings, not depending from the sensory nerve endings. Either can be axons or dendrites. It depends that the information, the signal is entering the cell body from the periphery, giving information what is happening around the organism, what is happening inside it. Here we have the Merkel discs composed of the Merkel cells and the axon terminals within the skin, epidermis, dermal junction. Here you have the Meissner corpuscle, here you have the Pacinian corpuscle. However, the Pacinian corpuscle is much bigger, so it is not in the scale in comparison with Meissner corpuscles of the dermis of the skin. This is the free nerve endings. Here you have the Ruffini corpuscle. Here you have the Krause end bulb, the muscle spindle, and Golgi tendon organ, which are approached. The Merkel cells, each associated with expanded nerve endings, form the Merkel disc, which function as tonic receptors for sustained light touch and for sensing of the object's texture. Prenerve endings in the papillary dermis and extending into lower epidermal layers of skin, as well as the cornea of the eye, which respond primarily to high and low temperatures, pain and itching, but also function as the tactile receptors. Root hair plexus is a web of sensory fibers surrounding the bases of the hair follicles in the reticular dermis that detects movements of the hairs. Here is light micrographic representation of the free nerve endings traveling from the connective tissue to the stratum basalis of the epidermis, and sensing the touch or itching of the segment of tissue. Meissner corpuses are elliptical structures, 30 to 75 micrometers by 50 to 150 micrometers, consisting of sensory sensory axons, winding among flattened Schwann cells arranged perpendicular to the epidermis in the dermal papilla. Meissner corpuscles initiate impulses when light touch or low frequency stimuli against skin temporarily deform the shape. They are numerous in the fingertips, palms, and soles, but decline slowly during aging after puberty. Lamellated Pacinian corpuscles are large oval structures, approximately 0.5 millimeters to 1 millimeter in diameter, found deep in the reticular dermis and hypodermis, with an outer capsule of 15 up to 50 thin concentric lamellae of flattened Schwann cells and collagen fibers surrounding a highly branched unmyelinated axon. Pacinian corpuscles are specialized for sensing coarse touch pressure, sustained touch, vibrations with distortions of the capsule amplifying the mechanical stimulus to the axonal core when the impulse is initiated. Here you have the light microscopic view of the Meissner corpuscle in the papillary layer of dermis. Here you have epidermal stratum basalis. Here is this similar Meissner corpuscle in the stratum papillaris of dermis with silver stained impregnation method. However, it is magnification is smaller because the size of the Meissner corpuscle is similar here and here, so that this magnification is much smaller than the left-hand corner magnification. Pacinian corpuscle, or lamellar corpuscle of Pacini, connective tissue, lamella, outer capsula, inner capsula, consisting of the flattened attenuated fibroblasts, and centrally placed non-myelinated nerve fiber terminus, which is taking the stimuli of the touch and pressure. Sensory nerve endings, Pacinian corpuscles are also found in the connective tissue of organs located deep in the body, including the wall of the rectum and urinary bladder, where they also produce the sensation of pressure when the surrounding tissue is distorted. Krause and bulb are simple, encapsulated, avoid structures with extremely thin, so collagenous capsule penetrated by a sensory fiber. They are found primarily in the skin of the penis and clitoris, where they sense low-frequency vibrations. Ruffini corpuscles are collagenous fusiform capsules anchored firmly to the surrounding connective tissue with sensory axons stimulated by stretch, tension, or twisting, torque in the skin. Muscle spindles and Golgi tendon organs, both are sensory proprioceptors or proprioceptors, in which sensory dendrites wrap around trafusal fibers in small specialized fascicles or around myotendinous collagen bundles, respectively. Here you have another example of Pacinian corpuscle, lamellar corpuscle of Pacini in the stratum reticularis of skin. Here you have the muscle spindle consisting of the intrafusal muscle fibers, small fibers surrounded with the axon terminal, sending the impulse of force of contraction or force of stretching the Golgi tendon organs, sending to the central part of the reflex arc, to the cell bodies of the sensory neurons, either in the spinal cord or in the spinal ganglion. Neurons communicate with other neurons or with effector cells by means of synapses, specialized junctions between the neurons that transmit impulses from one neuron to another or to a effector cell, muscle or gland. Typical synapse includes presynaptic knob, the end of the neuronal process from which neurotransmitter is released, synaptic clefts, 20-30 nanometer space in between presynaptic knob and postsynaptic membrane, and that is postsynaptic membrane on which are located receptors for neurotransmitters. In most of the cases is, for example, acetylcholine. Synapse between an axon and glandular cell is termed neuroglandular junction. Synapses between axon and muscle fiber or cell is termed neuromuscular junction or motor end plate. Structure of these junctions and the transmission of nerve impulses across them are similar to those of neuron-neuron synapse. So here you have the different type synapses, the axodendritic synapse, axosomatic synapse, axodendritic with through the spine apparatus of the dendrites, and here you have axodendritic synapse in increased magnification with presynaptic membrane consisting the synaptic vesicles, neurotransmitter accumulated synaptic vesicles, the synaptic cleft, the postsynaptic density, which is consisting of the receptor sites for the neurotransmitters. Here you have another illustration of the motor end plate or effector nerve endings with myelin sheaths ramifying to terminal processes or motor end plates. Here is the presynaptic pole with synaptic vesicles filled with neurotransmitter. Here is synaptic cleft, postsynaptic membranes with invaginations of the postsynaptic membrane containing the receptors in postsynaptic density, and the invaginations forming increasing the surface of the plasma membrane of the target skeletal muscle fiber. This is a myofibril consisting of actin and myosin filaments. These are mitochondria, and this is effector nerve endings or motor end plate in composition. Here are the motor end plates in the light micrographic representation with ramifications of the axon terminal. Here you have the presynaptic pole with synaptic vesicle, synaptic cleft, postsynaptic membrane with receptors or within the postsynaptic density receptors for the neurotransmitters. Signal is coming from presynaptic pole to the postsynaptic membrane only in one direction. It is called the physiological polarization of the reflex arc, which is basic characteristic of the nervous system arrangement function. And few words about development of the nervous system, which will be the end portions of today's presentation. The nerve tissue develops from the neuroectoderm in the process called neurulation, beginning from the 18th day of prenatal ontogenesis. Neural tube is formed till the end of the 4th week from neural folds approaching each other and gives birth to organs of central nervous system. Approximately 28 days, 3 primary brain vesicles, forebrain, midbrain, and hindbrain develop. In the 6th week embryo, they transform into 5 secondary brain vesicles. Neural crests located on both sides of the neural tube beneath the developing epidermis give birth to peripheral nervous system. Spinal and autonomic ganglia are developing from neural crests in parallel with the development of the spinal cord. Here we have the designated remarks for the neurosystem neurulation during the 18th day of embryonic development, 19th day, 20th day, and the 23rd day. I am showing you, emphasizing your attention of the neural tube, neural crest, notochord, somites, notochord, neural crest, neural tube, and superficial located developing the epidermis of dorsal ectoderm. And here is a lateral view of central nervous system on the week 4th and week 6 of embryonic development, including forebrain, midbrain, hindbrain, and the spinal ganglion and spinal cord development. Here you have the telencephalon, diencephalon, melencephalon, metencephalon, pontine flexor, melencephalon, and primordial spinal cord developing. Neural plasticity and regeneration. Certain regions of the central nervous system, such as near the ependyma, retain rare neural stems and progenitor cells that allow some replacement of neuronals throughout the life. Neural plasticity involving formation and remodeling of synaptic connection is also prevalent throughout life. The complexity and distance of the neuronal and glial interconnections with the central nervous system makes regeneration and restoration of function within this tissue after major injury very difficult. The more simply organized peripheral nerves have better capacity for axonal regeneration, a process involving the reactivation of the perikaryon, Schwann cells, and macrophages. On the next slide, you see the site of injury, which is leading to the degeneration of the nerve fiber and myelin sheath. The atrophic changes of the muscle is becoming thinner unless it gets the signals from the neurons, which is giving the neuronal support of the function of the muscular tissue. The Schwann cells are proliferating and restoring the axon permeability, developing the root from which the axon will further grow and enter the end plate of the neuromuscular junction. But it can be the cord of Schwann cells and disorganized axon growth. When it's not entering the muscle is becoming atrophic still more than under the regenerating process of the nerve tissue, neural tissue. And this is leading to the incomplete function of the muscular tissue. Here you have the examples of the nervous system pathologies, which you are supposed to look at home in the internet and compare with the normal nervous tissue histology, which was discussed and I presented to you right now. This is the end of the topic, which I plan to present to you on this lecture. These are the recommended reading from the different sources for expanded knowledge of histology and embryology. And this will be the end of today's lecture. Have a nice day. Thank you for your attention. Goodbye.